The first speaker today is Michael Dudek, who did his bachelor's and master's and bachelor's at the University of Tübingen. And he then did a PhD in immunology at the Technical University of Munich. And he's now a postdoc there in the Institute of Molecular Immunology. And he is studying autoimmunity in the liver and other organs. So today, Michael's going to talk about the newest instrument from Nanin, which is the AtlaZ, which is an impedance platform. So this started off back in the old days, label-free GPCR signaling. Then people realized you could use impedance to also make measurements of spontaneous contractility of stem cell cardiomyocytes. So that moved impedance into another big area of drug discovery, which is cardiac safety pharmacology. And now we have these new innovations on the platform, which allows the use of this for study of immuno-oncology and CAR-T. So this is what Michael's going to talk about today, using the AtlaZ to study cytotoxic T cells. So I don't know very much about this, so I'm looking forward to learning from Michael's talk.
So, hello, everyone. Everyone finally made it. So everyone can hear me? Yes or no?
Yes.
All right. So yes. Yeah, thank you very much for the kind introduction. Let me go back. Yeah, for the kind introduction, I'm currently a postdoc in Professor Knolle's lab at the Institute of Molecular Immunology, and this is basically my focus. It's immunology and in specific autoimmune diseases, which just evolved from my PhD project, and where I heavily use the competitor machine, the Xcelligence device, and you will see some of these results here in this presentation, but also some results with the new device, the AtlaZ platform. And I will give you some insights into my project, and also some insights of some other data from collaborators regarding cancer immunotherapy. So just to be everyone on the same page, a brief introduction into immunology and which immune cells harbor cytotoxic functions. You have on the left-hand side the innate arm of the immune system, and on the right side the adaptive arm. And you have many immune cells, but some of these are equipped with cytotoxic capacities, and these are mostly granulocytes, T cells, and mast cells. But they have distinct functions because they predominantly localized in different areas. And this is for granulocytes and mast cells, mainly in the circulation, the blood, and the immune cells that I'm more interested in the T cells are experts in the defense against pathogens in the tissue. And like a basic fundamental concept on how T cells are eliminating infected target cells is through MHC class restricted antigen recognition and killing of cells. So in principle, you have an infected cell, antigens are presented on the surface, and this is like the signal for the T cells to specifically kill only this infected cell and leave uninfected tissue intact. So that's why this immune system and immunology is so fascinating, because we have this kind of system, and it eliminates tumor virus-infected cells. But, and this is in a harmful way, during autoimmune diseases, it can also lead to elimination of uninfected cells. Then on the other hand, you have NK cells that eliminates target cells through a different mechanism, where you have, for example, immune evasion by tumor cells or infection that downregulates the MHC molecule on the surface, and this is then triggering NK cells to mediate immune attack. And in the middle of it, by purpose, and this is something I was able to show 2 years ago, which was published, that there's also another form of immune attack of T cells, and this goes through an MHC unrestricted manner. So here, a release of signals from a target cell is able to activate the T cells and eliminate the target cell. And so far, it's not really known what the physiological relevance of this mechanism is, but we know that during autoimmune diseases or like inflammatory conditions, it can also eliminate healthy cells. And this is a novel concept, and this was not new so far for this kind of immune cell cluster. And to get a bit more insights on how T cells can eliminate target cells is that it can either be through membrane-bound molecules like FAS-L, FAS-ligand, and TRAIL, or through intercellular weapons like perforin and granzymes. And these have to be also be in check by inhibitory mechanisms, and this brings me to also to immune therapy for cancer and also to autoimmune diseases, because if these checkpoints are not intact or like overexpressed, then the immune system is not functioning properly. And here we have also membrane-bound receptors on the T cells, could be PD-1, CTLA-4, and it could also be soluble cytokines like TGF-beta and IL-10. And it could also be other means, like if you have in the tissue lower oxygen levels, you have depleted nutrients, you have an acidic environment, or you even have increased levels of potassium. All of them together can inhibit the function of T cells. And this is especially true for in a tumor microenvironment, but it could also be that if the inhibitory mechanisms are not intact and in place, then it can also induce immune reactions that are not wanted and leading to autoimmune diseases. So I divided my talk presenting data from these two main areas, tumor and autoimmunity. So for the tumor-related data, the basis for that is that over the years there happens a revolution in cancer immune therapy, because we have new possibilities to eliminate tumor cells in the body, and this is through the application of immunotherapy. And this is done by two ways: either using antibodies that unleash the effector function of T cells, blocking the inhibitory axis, or engineering T cells in a way that they are highly specific for tumor cells, and which express abundant tumor antigens on the surface, which can be engineered to be specifically targeted by these T cells. And this really revolutionized this area, and also the need for quantification and quality check of these products, clinical products, are important. And that's why there is also a very high need for a specific devices that can monitor the killing capacities of these T cells. And I just figured out that the y-axis should be percent of death cells and that percent of cytolysis. So here I just created 4 different pictures how the cytotoxic assay could look like, and to give you a feeling why time resolved and kinetics of T cell mediated killing is so important to interpret data, and also for the clinical or has clinical relevance. So here I created 4 circles that shows that when you only pick one time point, it gives you basically the same result. But if you have many time points, then you can have a better interpretation of your product and your T cell. For example, looking on the left corner, if you have like an exponential killing of tumor cells, this is fast and efficient, but at the same time, it can also raise concerns because it might be too strong in the body and creating possible side effects, for example, a cytokine storm. There's also the possibility that you have first a slow and then an efficient killing. It could be the best compromise for this product, for the engineered T cells, because in vivo this would be enough to kill tumor cells, but for some certain tumors entities it could be not efficient enough. Then we have the curve where we first initially have a fast killing, but in the end an inefficient killing, because clearly you see a rebound of the target cell, the tumor cells. So here we have the danger of outgrowth of remaining tumor cells. And the fourth possibility, that first you have a delay, but then an efficient killing. So it could be for some tumor cells good to have this kind of a curve, but for aggressive tumor cells which outgrow very fast, it could be, yeah, not the best option to have this kind of a T cell product. So that's why the value of such machines that monitor over time the killing capacities is very fundamental to understand the dynamics, and then also correlate this data with the clinical data from patients that receive these products. So coming then to the real data, and here this is like one of the initial experiments that we have done with the AtlaZ system, that we have here an allo system where we applied T cells in different effector to target ratios to a lung epithelial tumor cell line, and you can clearly see that the more T cells you add, the better the killing capacity is, and the less likely is that the tumor cells rebound. And this is data from a collaboration partner from the Andreas Schmitz from Rechts der ISA, that checked their CAR T cell, which has a specific TCR against a EGFR receptor on tumor cells, which are abundantly expressed. And here also the same picture, the more you add 3 to 1, the better and efficient killing of the tumor cell you have. And this might vary between tumor cell lines, because every tumor cell line is different, and it will be very important to have this kind of readout to check the number of T cells that needs to be infused in patients are important. This method, or this device, is also in a way important because it also gives you hints whether these products creates a resistance against a tumor. So here, this is from a publication where they checked that a specific receptor on these T cells, if this is knocked out, lead to worse killing capacity of these cells. So meaning it acquires resistance from time to time, and knowing resistance mechanisms on these products, and in the end eradicating these resistance mechanisms, would lead to an optimized CAR T cell product, and also leading to an efficient cancer immunotherapy. So this was the part for the tumor, and now coming to the part that my expertise is lying in, is like autoimmunity. And I already pointed out in the introduction, we discovered published 2 years ago a novel concept of how T cells can eliminate target cells, and this is through an MHC unrestricted manner. And here this is in a more detailed view, which might be also interesting for the upcoming speaker talking about P2X receptors. Here we discovered that P2X7 is an important receptor which senses extracellular ATP released from stressed immune cells, stressed hepatocytes in NASH patients, that triggers calcium influx in these cells and activating cells, and inducing the effector functions. And this is what we have published in the mouse, in the preclinical mouse data. But what this current ongoing project is whether this also applies for human CD8 T cells, like autoaggressive human CD8 T cells, and whether we have similar mechanisms here or not. And I can give you the answer there are not dissimilar mechanisms, so this is something different in the human setting. Here's still already still raw data from the mouse system. So we performed, these are exogenous data, experiments where we had murine hepatocytes and adding our cytotoxic T cells and blocked either the P2X7 receptor on the T cells or the release of ATP through pannexin inhibitor from hepatocytes. And this worked here. And to show you the difference between autoaggressive CD8 T cells and antigen-specific T cells is that when you block these two pathways in this setting where T cells recognize an antigen and eliminate quite fast the infected hepatocytes, then these mechanisms are not really functional. So meaning we have activation access in these cells that this can discriminate between autoaggression and antigen-specific killing, which was at the time very astonishing and new, and which motivated us to check whether we find similar differences in the human setting. And in the human setting, it looks a bit different. So here again, exogenous data, where we add human hepatocytes on the bottom and check the killing dynamic of autologous CD8 T cells stimulated with IL-15 or allogeneic CD8 T cells stimulated with IL-15. And you can see that there's not much of a difference between the killing dynamic of these two cells, showing us that also the autologous IL-15 stimulated CD8 T cells are very powerful and efficient in killing either even autologous hepatocytes. And these are data from the AtlaZ system. When we compare, like when we see these HLA deficient target cell lines, so this is also tumor cell line, but are deficient for HLA molecules, and checked the killing dynamic of IL-15 stimulated CD8 T cells with like the prototypic killers and NK cells, then there's also not so much of a difference, showing us that these T cells are really very potent in eliminating target cells. And this was really striking to us because this was not really expecting, which we have not really expected. And this is something I can't disclose right now. So what we also found out is that we found a specific inhibitor that can block this specific killing machinery autoaggression. So by other experiments that we have performed, we could find an enzyme in a signaling cascade of this T cells that when we block it, then we can prevent autoaggression. And this was specific for autoaggression because when we again go back into our also allogeneic system and use this inhibitor, then it was not effective here, showing us that we have here again an inhibitor or a regulator that can discriminate between these two cytotoxic mode of action. And also in case killing is not affected. So we think that having inhibitors that can selectively inhibit autoaggression compared to antigen-specific killing, because antigen-specific killing is critical for eliminating infected target cell, would be very important to find and also helpful for treatment of patients with autoimmune diseases. So this brings me to my summary. So in general, what I have showed you is that first of all, we need time-resolved longitudinal cytotoxic assays to really fully assess the killing dynamics and capacities of cytotoxic immune cells in general. So it's not on T cells, but can also be applied for other immune cells, and that it's helpful to really have the best interpretation possibilities of our killing immune cells. And like specifically data, what I have shown you is that inhibition of P2X7 in murine CD8 T cells was effective for the prevention of autoaggression, but ineffective for antigen-specific T cells. And so far, what we can see is that in the human setting, autoaggressive CD8 T cells have similar efficiency in rapidly killing target cells like allogenic T cells or NK cells, which was very surprising for us. And we can find them also in elevated numbers in patients with autoimmune diseases, which makes this topic very, very relevant. And yeah, I want to thank several people, of course, my supervisor, Percy, and also some of my PhD students that perform these experiments also on the new AtlaZ system, which we are really looking forward to expand our expertise with this machine and also expand our results with it. And also Andrea Schmitz for providing some preliminary data on CAR T cells. And I want to thank you for your attention. And yeah, I'm happy to take your questions and suggestions.
All right, thank you very much, Michael. We've got time for 2 questions. I've got a couple online, but I'll see if there's anybody in this room or next door. Next door, you've got to come in to me and speak into the microphone if you do have a question. We've got one on the front row, sir.
Yes, well, very interesting talk. I have to reveal my ignorance about immunology and oncology, but I would like to have an explanation for what you mean by normalized cell index. Could you sort of specify the signals that you reported? Maybe I've missed it, but what is sort of the cumulative message of this index?
Exactly. So the normalized index, cell index, means that we have, so at a certain time point, I set the cell index that is measured as 1. So this is the normalization. And because some of the curves, if you have not the normalized cell index, goes up and some of them down, but to have like every line and on the same page, then I artificially normalize these values. So normally, if you start to add these cells and have the absolute cell index, then it typically reaches cell index of number 2 or 3, and then this is the starting point where I normalize, and then it gives you the number of 1.
I understand normalization, but maybe the index escapes me.
And this is at 10 kilohertz here, like the measurement was running at 10 kilohertz, and this is like the impedance of this machine that gives you the output.
Maybe I can also help out here with a little bit of explanation. So, for example, if you have like 100% confluency in your well, this could give you like a cell index of 1. And then if you reduce the viability of the cells to 50%, you have like 0.5. So this is like a representation of sort of confluency in some cases, viability, cytolysis.
Yeah, thank you, Alan. Yeah, and Sonia, that's correct. Yeah, it's like always we start over with 100% confluency of each target cell line.
Whoa. All right, one more question, seeing as this is an ion channel focused meeting. There is quite a number of other ion channels involved in cancer. So have you looked, or are you aware of anything other than P2X7 in your cancer research?
So the P2X7 receptor that we discovered was on the T cells. So at that time it was pretty new that there is like kind of this receptor important for a function of T cells. But to be honest, this is what we have found so far very specific for the murine setting to being activated through ATP and P2X7. For the human CD8 T cells, we have not seen such an association. So that's why our focus is not so much on this ion channel on the human CD8 T cells right now, but there still exist also these ion channels on the T cells, but this is currently not our focus.
All right, thank you, Michael. That actually gives us a nice introduction to the nice speakers, so I thank you for that. So the next speaker is